This lecture is about membrane transport with a focus on the electrochemical gradient. The electrochemical gradient is one of those big picture concepts. And I'm going to go through a thought experiment with liposomes and then talk a little bit about the Nernst equation. Membranes are barriers to ions and other molecules, including proteins and nucleic acids. And it takes energy to get across a membrane if you're an ion, or if you're a big molecule, even such as glucose. Importantly, information is conveyed across membranes, and the electrochemical gradient is used by nerve cells to transmit electrical impulses from one part of the cell to another. So to understand how nerve cells work, we have to understand the electrochemical gradient. So let's set up a liposome experiment. Liposome is a membrane bilayer that's a complete sphere. Okay, so we just have water on the inside and water on the outside. We can prepare the liposomes with a little bit of salt on the inside and then transfer them to a lower salt concentration. So let's imagine that this is a liposome here, and we've prepared the liposomes in 100 millimolar potassium chloride. And then we transfer the liposomes into a lower concentration of potassium chloride. So we have 10 millimolar potassium chloride on the outside. Cells, real cells, have high potassium on the inside and low on the outside. So this is kind of like a real cell. So let's consider a series of questions. We're going to go through these questions in class, and I'll just preview for them here to help you understand. So in this experiment, what will cross the lipid bilayer, if anything? Consider that there's water on the inside and the outside of the vesicle, or the liposome. And what we have to think about here is osmotic force, because water does go across membranes at some slow rate. And over time, water will want to push in towards the higher salt concentration. Let's add an ion selective channel. This is a membrane protein that crosses the membrane bilayer, and it only lets in potassium ions. K+ potassium ions are the only things that can go through. So now, because we have a channel, potassium can go through. And there will be a little bit of water as well. Over time, potassium comes out, creating a net positive charge on the outside, because all of the K+ ions, the potassium ions, are building up outside. So the chemical force of a higher concentration of potassium chloride inside versus outside drives the potassium outside. However, an electrical force builds up because the chloride ions, which are negatively charged, are not transported by the ion selective channel. And in fact, the chemical force created by the higher salt concentration inside versus outside is much smaller compared to the electrical force. Charges don't like to be separated. And so you only get a few ions going across. The electrical forces are very large compared to the chemical forces. So this causes a membrane potential, a difference in charge, to build up across the membrane. And only a few ions can cross before the charges are so separated that you have a big electrical force counteracting the movement of ions through this ion selective channel. This separation of charge on the inside versus the outside of the vesicle or liposome, the separation of charge across the membrane bilayer, creates a membrane potential, which is symbolized as V. V sub m for membrane potential. And the units are volts or millivolts. So the important concept here is that charges don't like to be separated. They want to go back, and positive wants to be attractive to negative. Charges don't like to be separated. Because the electrical force is large, the actual number of ions that move is very small. It's about 1/100,000th of the total concentration of ions. So you can calculate how many ions move by calculating 1/100,000th of 100 millimolar. By convention, we define the membrane potential as the inside versus the outside. And we have an equation called the Nernst Equation, which will tell us what the membrane potential will be. This was worked out by a guy named Nernst, and the simplified version is shown here. Later, I'll show you how this equation was derived for those chemists who like those kinds of things. So the simplified version of the Nernst equation is a constant, 58 millivolts, times the log of the concentration outside over inside. And since we have 100 millimolar inside, 10 millimolar outside, by convention, the membrane potential is the inside minus the outside. So the Nernst equation is the log of base 10 of the outside over the inside concentrations. So in this case, we have the concentration outside of 10, the concentration inside of 100. So that's the log of 0.1, which is -1. So the Nernst equation tells us, and this can be measured, that in this situation, the membrane potential is -58 millivolts. That's for potassium. So now let's add sodium chloride to the experiment. Prepare the liposomes in 100 millimolar potassium chloride plus 10 millimolar sodium chloride, and then transfer them to a different buffer that has low potassium, like before, and high sodium on the outside. This situation is like real cells, because real cells have high sodium on the outside and low sodium on the inside. So now, what will experience net movement? We have a balance of salt, 110 millimolar on the inside and 110 millimolar on the outside. So really, the osmotic force is not present here. Let's add the ion selective channel. As before, this is selective for potassium ions, K+. Now, what will experience net movement? As before, without the channel, ions don't move. But with the channel, only the potassium ions will move. Since it's selective for potassium, sodium ions won't move. And because a little bit of potassium ions move, a small amount of water will also move, but not very much, because the salt is balanced. So water is also present and creates a much smaller osmotic force in this situation. Now, let's add an ion selective sodium channel in addition to the potassium channel. So So this is like real cells, which have both potassium and sodium channels. This one is selective for sodium, Na+, and will transfer only sodium. So, what will experience movement? Water won't move very much, maybe little. Potassium will move. Chloride will not move because they're an ion selective. They're only selective for positively charged ions. So we'll have both sodium and potassium moving. Now, what is the membrane potential in this situation? Let's consider each channel by itself. If we erase the sodium channel and just have the potassium channel present in this situation, the Nernst equation tells us, as before, that the only ion to consider, because the only ion that's moving, is potassium. So the Nernst equation is the log of potassium outside over potassium inside minus 58 millivolts, as we calculated just a second ago. So let's erase the potassium channel and consider just the sodium channel in this situation. Now, the Nernst equation tells us that the outside is higher, 100 millimolar, over the inside, 10 millimolar. So it's plus 58 millivolts. That's just with the sodium channel present, because the potassium channel is not present right now. So let's put them both together. Now, what's going to happen? Both channels are open. Sodium can go in because its concentration is higher on the outside. Potassium can go out because its concentration is higher on the inside. At steady state, the inward and outward currents match. So what will the resting potential be? This is called steady state. Ions are flowing, but the currents will match because of those electrical forces. The resting potential, and you can derive this quantitatively, depends on how fast the ions flow through these channels. That is the relative conductance. The conductance means how fast the ions flow. So if the sodium ions are flowing faster, then more of those will be inside, and you'll have a net buildup of positive charge inside. Conversely, if the potassium ions are flowing faster, you'll have a net positive charge built up on the outside. So just in this simple experiment, you can see you can change the membrane potential from minus 58, the potassium, to plus 58, the sodium channel, simply by changing the relative conductance of the channels. Or, put another way, you can change the membrane potential this drastically just by opening and closing channels. And this is how nerve cells work, as I'll show you in the next lecture. So the relative conductance dictates the membrane potential. That is how fast the ions flow. And so if you open channels selectively, you can set the membrane potential between very negative to very positive. That's how the action potential works. And I'll show you that in the next lecture. Channels are membrane proteins that have closed and open conformations. So in the closed state, they don't let any ions through, and in the open state, they let ions through. And they're selective, actually selective for specific ions. That has to do with how big the ions are. The way they open and close can be through a conformation change that's regulated. And in the case of the action potential in nerve cells, the membrane potential the difference in charge across the membrane actually dictates the conformation of the channel between open and closed. There are other ways to open and close channels too. Channels can be phosphorylated or activated by ligands. So this gives us a way to transmit information across the membrane. And in a nerve cells have the cell body and a long part called the axon, which ends up at the nerve terminal. Cells are usually inside negative, but by switching these channels transiently, you can have a small patch of membrane where the membrane potential switch it reverses. So the membrane potential reverses to become, instead of inside negative, it becomes momentarily inside positive. It's important to remember that changes in the membrane potential involve ion flow, but it really doesn't appreciably change the concentration inside the cell. Only a small number of ions flow. So the concentrations are maintained, but the membrane potential can switch from inside negative to inside positive. The electrochemical gradient is important for a number of things in addition to nerve cells. It's a way to store energy. You can think of it as a kind of battery. So the asymmetric distribution of charges is a way to store energy. And that's used in addition to nerve cells in mitochondria and chloroplasts. When we talk about those, you'll see that the electrochemical gradient is used to store energy to drive ATP synthesis. This amazing machine, the ATP synthase, will use the concentration gradient of protons to drive this motor which makes ATP. That happens in mitochondria and chloroplasts. And we'll talk about that when we talk about mitochondria and chloroplasts. So next, I'm going to tell you about the Nernst equation, and I'm going to derive it for you. And this I entitle diversity because I understand that people have diverse backgrounds, and so this part is really not for the test, but for those people who want to know about chemistry. The Nernst equation in its full form is shown here. Membrane potential is RT, the gas constant, times the absolute temperature, over the charge, Z, the valence of the ion. So sodium and potassium have a valence of 1. And the Faraday constant in the denominator as well. And this one is the natural log. That's different than the log base 10 that I showed you before. So how is the Nernst equation derived? It starts with the chemical forces and takes into account the electrical forces. And we can separate those. So for ions moving across a membrane, delta G is negative. Okay? The chemical forces are driven by the difference in concentration. Concentrations want to equalize. Okay? So the free energy change for 1 mole of ions moving across a membrane is given by this equation from gas law. Delta G is negative RT times the log, natural log, concentration outside over concentration inside. This creates electrical forces because charges don't like to be separated. And that can be defined, the free energy, as delta G volt, the voltage difference. And that is just simply this valence times the Faraday constant times the membrane potential. That's from electrical theory. At equilibrium, the two forces balance. So the chemical forces want to drive one way, the electrical forces the other. So they sum to 0. Okay, so this plus this equals 0. Or another way to put it is this equals this. So we can use this and substitute these equations here. So this is the same version, this is the same equation as this, just giving these substitutions. So you can rearrange the equation and solve for the membrane potential. And that's how you derive the Nernst equation, shown in the logarithmic form. Or if you want to convert it into log 10, at room temperature for a univalent ion, that charge is 1, Z equals 1. At 20 degrees, you come up with this number, 58, for the constant RT over ZF. That's how the Nernst equation is derived. In the next lecture about membrane transport, I'll show you how the electrochemical gradient is used for nerve cells and for transport of other molecules such as glucose. Thank you.